This is a revision class for you in BCH 418. First of all, you need to know the difference between paralogous gene and orthologous gene. You must know the difference. Like I said, paralogous gene and orthologous gene are two types of homologous gene. So what is a homologous gene? A homologous gene are two sets of genes that share the same ancestor, or they originated from a common gene. Okay, so when two sets of genes originated from a common gene, you call those two sets of genes homologous genes. All right, now there are two types of homologous gene. They are paralogous gene and orthologous gene. So what is the difference between the two of them? Gene is said to be paralogous if they were separated by gene duplication. All right, two genes are said to be paralogous if they are separated by gene duplication event. The underlying word there is gene duplication. So if a gene in an organism is duplicated to occupy two different positions in the same genome, then the two copies are paralogous. Paralogous genes often belong to the same species, but in some cases it's not usually the case. Example of a paralogous gene is the hemoglobin gene of humans and the myoglobin gene of a chimpanzee. Okay, they are homologous genes that originated from the same ancestor. Okay, as a result of gene duplication. Function may be retained, or they may have changed function due to the duplication. Okay, so this is about paralogous. But what about orthologous? If two genes are separated by a speciation event, two genes that have common ancestor, in other words, the two genes are homologous genes, but they were separated as a result of speciation when evolution took place, and the gene now formed two species. Those two genes are said to be orthologous. So orthologous or orthologous genes are genes in different species that originated by vertical descent from a single gene of the last common ancestor. For instance, the plant flu regulatory protein is present both in Arabidopsis and Chlamydomonas. So this is an example you will give for orthologous gene. The key point you need to take home here is a homologous gene is a gene inherited in two species from a common ancestor. Right? Orthologous genes are homologous genes when the gene diverges after speciation event, but the gene and its main function are conserved. Now, if a gene is duplicated in a species, that's now look at the underlying word, duplicated in a species, the resulting duplicated genes are paralog of each other. Did you get that? So then homolog versus analog, analogous traits and homologous traits. Like we said earlier, homologous genes are common two genes that originated from one common ancestor. Analogous genes are genes that do not originate from a common ancestor, even though they look similar in function, but they do not originate from a common ancestor. So you say it's analogous. All right? Example: human and chimpanzee have similar fingers because they share a common ancestry. So those fingers in human and chimpanzee are homologous. All right? Dolphin and shark both have pointed dorsal fin on their back, but this isn't homologous because they are not of the same ancestor. So you can look at it here. So this one is analogous analogous structure, same function, differ in fundamental structure, different ancestry. Example, wings in bat, birds, and insects. So when you see wings in bat, birds, and though they have the same function, but they are not of the same ancestor. They have different species altogether. So you say they are analogous. But homologous structures are those ones that are of common ancestor. So you can see, like the hand of a human being, the hand of a cat, the hand of a whale, the hand of a bat. They are all of the same ancestor. So they are homologous structures. So we say give an example of orthologous gene in orthologous gene. So orthologous gene are those which arose from a common ancestor gene. Example, the gene for the mouse enzyme trehalose. Alright, we have an ortholog in human that also make the trehalose enzyme. So the trehalose enzyme gene in mouse is an orthologous gene also in human being, the human trehalose enzyme. Then you have an example of paralogous gene is the gene for hemoglobin in human and myoglobin in chimpanzee. Please take note of the example. It's very important because the question will be explain orthologous gene, paralogous gene with specific examples. Alright? So both humans and chimpanzee share common ancestor. So that is why the gene for hemoglobin and human myoglobin are paralogous gene. Okay? So that's the example. Then xenolog or xenologous genes are those genes that are found in different species through horizontal transfer of DNA or genetic material. Alright, they are a subset of homologous gene as well. They most occur usually in prokaryotes. What's an example of homologous gene? An example of xenologous gene or xenolog is the bio4 gene, which is horizontally transferred between many species. This bio4 gene is the gene for the synthesis of biotin. So this is a summary of it. You can specifically this is a summary. Homology, homologs, ortholog, paralog, xenolog, horizontal, lateral gene transfer. Please take note of it. Then, when we talk about approaches in genomics, we have comparative genomics and then what? Functional genomics. So they can ask you to explain functional genomics and comparative genomics. Please, once you want to explain comparative, just say comparative genomics is a large-scale holistic approach that compares two or more genomes to discover the similarities and differences between the genome and to study the biology of the individual genome. What is it based on? What is the basis of comparative genomics? Evolutionary biology and phylogenetic reconstruction of the tree of life, drug discovery program, function prediction of hypothetical protein, identification of genes. So this is the aim or the focus of comparative genomics. Then what is the difference between comparative genomics and functional genomics? The main difference between functional genomics and comparative genomics is that functional genomics focuses on understanding how genes and their products function within an organism, while comparative genomics aim to uncover evolutionary relationship and genomic differences between organisms. See? So functional genomics is a field of molecular biology that attempts to make use of what? Vast wealth of data. So if I ask you to explain functional genomics, just explain this thing in blue now. Functional genomics is a field of molecular biology that attempts to make use of the vast wealth of data produced by genomics and transcriptomics to describe gene and protein function interaction. What is the focus? What is the aim of functional genomics? It focuses on the dynamic aspect such as gene regulation, transcription, translation, gene expression, and protein-protein interaction. It attempts to answer the question about the function of DNA at the levels of genes, RNA, transcript, and protein product. So please, this is specifically because the question can be explain functional genomics and the aim, and state the aim of functional genomics or the focus. Please take note of that. Very, very important. So we haven't said that. We talk about the gene sequencing, which is the approach in genomics. One of the approaches in genomics is gene sequencing. So you need to understand this, the Sanger sequencing and the next generation sequencing. It's very, very important. No one talk about gene sequencing. We are talking about the process of determining the number and the position of each nucleotide in a gene. Okay? So if it's in genome, you are talking about the entire genetic makeup of an. So determination of the number and the position of all the nucleotides in the entire genome of an organism. That is genome sequencing. So there are two major approaches that are used: the Sanger sequencing and next generation sequencing. You need to understand what is the principle. In principle, the concept behind Sanger and next generation sequencing technology are similar. In both next generation sequencing, that is NGS, and Sanger sequencing, also known as dideoxy or capillary electrophoresis sequencing, in both of them, DNA polymerase adds fluorescent nucleotide one by one onto a growing DNA template strand. Each incorporated nucleotide is identified by its fluorescent tag. Okay, so this is the similarity between the two of them. Their principle it has to do with both. Involves fluorescent nucleotides being added by DNA polymerase onto a glowing template strand. So the fluorescent tag is what enables you to identify the nucleotides. But the difference between the two, Sanger sequencing and NGS, is based on the volume. Whereas the Sanger method only sequences a single DNA fragment at a time, NGS is massively parallel, that is sequencing millions of fragments simultaneously per run. So the process translates into sequencing hundreds to thousands of genes at one time. So NGS also offers greater discovery power to detect novel or rare variants with deep sequencing. What is the advantages of NGS over Sanger sequencing? Advantage of NGS include they can screen more samples cost effectively, detect multiple variants across regions of the transcriptome, increase sequencing depth, mutation, resolution, sensitivity, and discovery power, high sensitivity to detect low frequency variants, faster turnaround time for high sample volume, comprehensive genomic coverage. So all these ones are very higher capacity with sample multiplexing, ability to sequence hundreds to thousands of genes or gene region. See the advantages. So next generation sequencing, what's the principle? Like I said, the principle is a type of DNA sequence that uses parallel sequencing of multiple small fragments of DNA to determine sequence. Now, what are the steps? If I just ask you steps in NG, very, very important. Please, if you are preparing for, say, 418 exam, you need to know these steps. As I've explained it here, look at them. Step 1 in next generation sequencing involves DNA extraction. Step 2, library preparation, which has to do with DNA fragmentation. After DNA fragmentation, you add the adapter, and that gives you a library, DNA library. So from fragmented DNA plus adapter sequence together, we make DNA library. That's step 2. Then step 3 is the sequencing proper, and then step 4 is the analysis. All right. So look at it here. The basic next generation sequencing process includes fragmenting DNA or RNA into multiple pieces, adding adapters to form the library, sequencing the library, and reassembling them to form genomic sequence. You can also use this one to say it. Number 1, library preparation. Number 2, DNA library bridge amplification. Number 3, DNA library sequencing. Number 4, alignment and data analysis. So you can just, if you want to draw, you can draw it like this. If they ask you to write an exam for, if you're able to draw it this way, you have to draw this thing now like this, you have a full mark for it. What about steps involved in Sanger sequencing? Steps in Sanger: number 1, denature double-stranded DNA using heat. Number 2, make multiple copies of segments. Number 3, attach a primer. Number 4, add 4 of the polymerase solution. The various dideoxy have dideoxy guanidine, dideoxy histidine, dideoxy thymidine, dideoxy adenine. So you add all of them. They are different. There are 4 of them. Dideoxy. So you then grow complementary chains until termination die. That dideoxy is the termination. Once you encounter the dideoxy, it will terminate. Then denature the grown chain. Denature means to separate them. Then electrophorese the 4 solutions. You see? So when you look at these two now, you see that the difference between this one, this Sanger, and the next generation sequencing is that in next generation sequencing, there is usually the process of library formation, DNA library, and also you see there is this sequencing cycle. This sequencing cycle in this step 3 here is not present here. Alright? So this sequencing cycle is what enables this particular process of NGS to sequence a large expanse of genes within a short time. You see, because you do it, you can use this process and sequence the entire genomic makeup of an organism by this sequencing cycle. But here in this Sanger, you will not see it there. You can't see sequence. So in Sanger, you have to sequence 1 gene fragment at a time. 1 gene fragment at a time. But here, you can fragment the entire genomic makeup of an organism and sequence all of them. So you can see it here. You can sequence all by this sequencing cycle. This step 3. This several times. So this one is the Sanger proper. You can see the Sanger steps involved in Sanger sequencing. Look at it. See? The steps involve single-stranded DNA with unknown sequence, which is denoted by the blue color. Add DNA polymerase. Add DATP. All these ones are the nucleotides. Deoxy ATP, deoxy TTP, deoxy GTP. When you add on, you now add radioactively labeled primer. That primer is what we enable the growing chain to start, the polymerase to attach and start synthesizing the growing strand. So all this one now, the dideoxy, where they will terminate. Each of the nucleotide has its own dideoxy, so that once the polymerase encounters any of the dideoxy, the chain will terminate. So with that now, you can have different fragments of it. So you can see step 3, step 2, then step 3 is gel electrophoresis followed by autoradiography. Then you now have the step 4, which is sequence of the templates, resequence of new strand, and then they do sequence of templates. So you can follow that process. Just write it that way in exam and you are done. Okay? So what is the difference between NGS and next generation sequencing? The critical difference between NGS is volume. While the Sanger method only sequences a single DNA fragment at a time, NGS is massively parallel. That is sequencing millions of fragments simultaneously per run. Okay? So with this, you have covered everything. So just take this slide as it is in this video and try to read it over and over again before your exam, and everything you need in your exam it will be there. So that's what you need to that's what you need. Thank you, and I wish you success.